Hello, Biology 100. This is Chapter 8, the first part of this lecture. We're talking about DNA. What is DNA? As you know, in our Kahoot, we learned that DNA is deoxyribonucleic acid. That's what it stands for. It's the genetic code for all life, the blueprint of our traits. So our DNA, right, is really our genes. And that is what produces our traits or our phenotype. DNA is a double-stranded molecule made up of nucleotides. The nucleotides are the monomers, or the single units of a piece of DNA. Each nucleotide is a sugar group, a phosphate group, and is one of four bases, A, C, G, and T. Here's what DNA looks like. Well, if you could see it, it's really microscopic, very, very small, can't even be seen under a microscope. You'd need an electron microscope even to see it down into the sausage shape. And so you can see it's a double strand. There's one strand, there's the other. It's wound up into a helix. And each of the nucleotides, a single nucleotide shown here, is base paired with another nucleotide on the other strand. And this pairing is very specific. We call that complementary base pairing. Complementary. Let me write that down for you. Whoa. Complementary base pairs, right? And complementing base pairs are always A with T or G pairing with C. And that's exactly what this says right here. We have A pairing only with T. So anytime there's an A on this strand, there's a T on this strand in that spot, and vice versa. So here's a T, that means there has to be an A. Here's another T, that means there has to be an A. Anytime we see a C, there's a G. Anytime we see a G, there's a C, CG, GC. And that's how those two strands, the single strand of DNA, is complementary base paired to the other strand to make this double helix structure right there. Hydrogen bonds, like we Kahooted, hold the base pairs together. So that's these. We call them H bonds or hydrogen bonds. So the DNA is kind of like a ladder, right? These would be the rungs of the ladder, except the ladder's all twisted up, so it'd be really difficult to climb. Why do we care about DNA? Because it holds our genes. Our genes are made up of DNA. A chunk of the double helix, right, might be a gene that codes for a protein that produces a trait, like one of the traits for your hair color or your ability to roll your tongue. The musculature to roll your tongue is from one single gene made up of DNA. If we have lots and lots of this DNA, right, a whole big long strand, that would make up a chromosome. So what part of the DNA represents the rungs of the ladder? Ooh, I don't know. Maybe it's the base pairs, right? So the rungs of the ladder are the H bonds, the hydrogen bonds between the bases. Here's what the bases look like. Okay, so this would be the base, right, thymine, or it could be a C there, or it could be a G there, it could be an A there. This is the sugar, that's the phosphate. What part represents the sides of the ladder? The sides of the ladder, which they're showing in blue, is really the sugar and the phosphate, and how they're linked together. So here's a phosphate, a sugar, a phosphate, a sugar, a phosphate, a sugar, phosphate, sugar. So this is the side, and then the bases stick in the middle, and they complementary base pair with bases sticking in the middle, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, on the other strand. So the two strands of DNA are held together by hydrogen bonds. So the question is the hydrogen bond that holds the base pairs together, remember there, remember there he is, right there, H bond, is that a strong or weak chemical bond? Well, you know, we skipped that chapter, that was chapter 2, right? But I'm going to tell you it's a weak chemical bond, but when there's lots of them, together they can make a strong association to hold those two strands together. The base pairing rules, like I said, A's and T's always go together, C's and G's always go together. Memorize this. These are going to be easy, easy, easy questions on the exam if you know the answers. Well, I guess any question is easy if you know the answers. If you memorize this, you're going to get lots of points. So when we know the sequence on one strand, because I've told you that complementary base pairing holds the two strands together, and complementary base pairing rules are A's always go with T's, and C's always go with G's, that means anytime you have a sequence of one strand, I'm just going to make something up here, what would be the sequence of the other strand? Well, if A's always go with T's, and C's always go with G's, and G's always go with C's, and T's always go with A's, and A's always go with T's, and G's always go with C's, you know the other strand. So on the exam, when they say the one strand of DNA looks like that, what does the other strand look like? All you do is write that, and bingo, points. The sequence of the nucleotide bases in the DNA or in a gene is what determines what protein is made, which determines what trait it is, which determines your phenotype. Remember, we talked about phenotype, or your physical manifestation is brought about by your genotype. What alleles do you have? Attached or detached earlobe shape? Those are alleles, all about earlobe shape. Right? Your sequences, the DNA nucleotide sequences, determines your genotype, determines what alleles you have, which then determines what traits you show, and that's your phenotype. So, if all genes are composed of just 4 nucleotides, how can different genes carry different types of information? Well, it's about the sequence. Which sequence mean? The order of nucleotides. How many A's, how many T's, how many G's, and how many C's, and in what order? A, T, G, C, 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 G, and its other strand, right? Might code for something completely different, or would code for something completely different, than a sequence at C, 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 A, G, G, A, T, C. Right, if we give us the complementary, which you should be able to do quickly because you guys know the rules, right? If this is part of a gene, that's going to give you a different protein and a different trait than this sequence, even though they're the same 4 nucleotides. They're in a different sequence, in a different order, different amount. Do you expect to see more variation in the sequence of bases between 2 members of the same species, or between 2 individuals of different species? Well, all humans have the same genes as each other, correct? But we could have different alleles. Let's say we're just looking at a gene from human A, let's say that's me, and human B, and let's say that's you, and we have the exact same sequence, look here and look here, TAC, TGC, AAA, uh-oh, I have a C and you have a T, and then TCA is the same, and ... meaning that's not your whole gene, right? There's lots more in there. But that's showing that we have one base pair different from each other, but I'm telling you it's the same gene. So if this is for tongue rolling, that might mean that we have 2 different alleles. It's still tongue rolling gene, but it's not the exact same sequence. It means it has to be a little bit different. So I'm a tongue roller. Tongue roll, yes. And if you have a little bit different sequence, but it's still tongue rolling, maybe you're no. You have the other allele. Chickens have tongues. Can they tongue roll? I don't know. Maybe they can. Have you asked a chicken? There's an experiment. Go ahead and do that. Let's pretend like they can. Or let's pretend like they can't. Let's say they can't, right? Why not? Because their tongue rolling gene is close to the same as ours, But look, it's got all these differences in it. Maybe those differences made it so no tongue rolling. The gene's there, but it doesn't work. It doesn't create the right musculature. So human being genes should be much closer together. The only difference is alleles. But between another species, that gene could either have a different function or not function at all, or just be different, right? Changes over evolution. So different alleles of a gene have the same DNA sequence or different DNA sequences. What do you think? What do you think? Has to be different, right? If it's a different allele, right? Different alleles code for different version of trait. Tongue rolling or not? Yes, no for tongue rolling. It's a protein that helps design the musculature of your tongue. If your tongue muscle is different than my tongue muscle, it has to have a different sequence in the DNA because your DNA codes for the RNA that codes for the protein that determines how well that muscle works. If it was the same sequence, it would work exactly the same. You'd either both be tongue rollers or neither be tongue rollers. The sequences have to be different to have a different allele, but not so different that it totally changes the function of the protein or makes a different function. That would be a totally different gene. So slight differences make different alleles. Well, what makes one allele different from another? See, right here. Yes, a little bit different. Do we see a difference? Where? Right here. Slight difference in the protein produced by the alleles, different trait version. It's still tongue rolling. It's just can you do it or not? Or it's still earlobe shape. That's the gene. One allele is attached. One allele is detached. They look different because some protein was slightly different that changed the shape during development. So how do we know? How do we get the sequence or the order of the DNA? Well, we have to sequence it. We read it. Whoa, almost puked. We can make many, many copies of this sequence and artificially replicate the DNA to figure out what the sequence is, right? We do this by mimicking how regular DNA replication works. Just like we copy, remember we copy DNA, we make the sisters, we go from the chromosome, right, to the 2, and then we condense it and it looks like a sausage. Remember, these 2 yahoos are the sisters. Copy DNA in S phase of interphase. Yes, I like what you've done here. Here's a DNA sequencer, right, making lots of DNA, and then we can figure out what the order is using really expensive equipment. And there you have it. If we're talking about the DNA differences between prokes and eukes, and just for kicks, let's do that, right? Remember, prokes have a circular chromosome, right? It is double-stranded, but it's just a circle. And then we know the eukes have the big chromosomes. If you're, a human, you have 46 chromosomes, etc. And so these are some of the differences between them. I'm not going to ask you specific questions about this chart, except that, right, the circle versus lots of linear chromosomes. And there you have it. There's part 1. Thanks, guys.